So it is my honor to introduce Dr. James Lee as our keynote speaker today. Dr. Lee, of course, PGY4 in the Department of Neurosurgery here at Mayo Clinic, Florida, and his lecture today will focus in human intracranial electrophysiology. So he obtained his bachelor's degree at the University of California in San Diego, and his medical degree at the David Geffen School of Medicine at UCLA in 2020. He then started his neurosurgery residency training here at Mayo Clinic, Florida. Some of the honors and awards Dr. Lee received throughout his career included Britain Alumni Leadership Scholarship in 2012, a UCLA Presidential Fellowship Award in 2017, and a Young Investigator Award by the American Epilepsy Society in 2021. He also received several scholarships, including the Mayo Clinic Diversity Scholarship in 2019. Here, we can appreciate Dr. Lee's contribution to research and his track record over the years, with over 40 publications and 490 citations on ResearchAid. So it's truly an honor, and we thank you for joining us today. So without further ado, Dr. Lee.
I just want to say a few words quickly about Dr. Lee. Over the last year, he's been doing research with me as well as Dr. Hermes up in Rochester on human electrophysiology. We've been very proud of the work he's been doing and looking forward to see what he shares with us. He's going to cover 70 years of electrophysiology based on his title, I guess. So we'll see, James.
1 year, not 70, but let's see. I can't see my screen here. All right, so my academic interests lie in how we process our visual experiences in our daily lives. And as a brief overview, the moment light stimulates our photoreceptive fields in the retina, the optic nerve sends downstream signals to the thalamus, then to our visual cortex. And although many scientists before us decades ago have made significant progress in research, we still have no clear understanding of how we process light, shapes, edges, lines, and colors into higher cognitive representations. For example, knowing when we see animals, cars, and people. And knowing exactly what we are seeing, I think it's incredible how we take in our visual experiences and form them into these lifelong memories. And we're able to process all of our visual experiences from the moment we open our eyes to shape our human experience. So this image shows a 3D image of the human brain and a better look at some of the visual cortex. V1 represents the striate cortex, where the input fibers from the retina and the lateral geniculate nucleus initially synapse onto the cerebral cortex. And beyond this area are V2, V3, V4. This is called extrastriate cortex, which is the concentric regions around V1 in an attempt to label areas where we presume are higher levels of visual processing. So David Hubel and Torsten Wiesel, they received a Nobel Prize in the 80s for their work in vision, and they inspired generations of neuroscientists. So we know that the signals in the retina and the thalamus respond to spots of light, but when recording in the visual cortex, the nature of these signals have like an interesting computational changes. With animal experiments, they show that within the primary visual cortex, the V1 that I showed earlier, we see responses only if you change the spots of light to edges of light, and if you move these bars of light in a certain orientation or direction. So from the primary visual cortex, how do we get these signals further propagate to form memories? And what do we know so far? So Kaiser and colleagues previously discussed the speed of light to argue that even images that present like 14 milliseconds, the neurons in the visual cortex of the monkeys are able to differentiate what type of images we saw. So this 14 milliseconds is very special because within these 14 milliseconds, maybe if you record from one neuron, you get maybe one action potential. So what does this mean? So this implies that when we see an image, the processing involves specific populations of neurons firing at once, instead of like the firing rate of neurons within a certain time frame. And with more advancements in technology, we're able to rarely record signals in the human brain. And in this paper, they show many different images to patients who have implanted electrodes for epilepsy monitoring. And in early visual areas, not the primary visual cortex, there seems to be neurons that specifically respond to categories of images. And it's pretty well known about the ventral temporal cortex that has areas that respond selectively to pictures of people. But what this paper showed me, interestingly, that there's also areas in the lateral occipital cortex, which is interesting in itself. And this categorical differentiation shows evidence that there's higher level of visual processing in the early areas. So before I present some of my figures, I'd like to thank Dr. Hermes and Dr. Miller in Rochester, who invited me to see their framework, protocols, and also allow me to work with their pre-existing data set. So in the existing data set I worked with had a similar task. So these patients with implanted electrodes saw 1,500 images, about 800 milliseconds each, which was a time sync to the neural data. And I wanted to reproduce the findings of the paper I just described by looking at early visual areas and see how the signals looked when patients saw many different images. And this is an example of raw voltage traces. I'm able to plot in the right lateral occipital areas. And here you see voltage differences each time the patient saw an image of a person, animal, or scenes. So just as an overview, how we calculate and analyze this raw data, let's say you have a raw voltage trace shown above, obtained from a patient. And the goal is to parse out these different oscillations of the trace versus time. So for example, let's say you have a trace that looks like the one shown above, then using computational methods, you can really parse out these different frequencies that represent different types of neural activity. So the traces I just showed you before, I can parse out frequencies usually between 10 to 200 and analyze these different frequency bands. And I particularly chose this high frequency band, which I define as 70 hertz to 170, because this likely represents the local neuronal populations in one electrode. A lot of complex steps are involved when you're processing this data. And some of these steps in itself can be like an entire thesis for a paper. For example, choosing between common average referencing versus bipolar referencing. It took me a while to really find the best method. So briefly again, choosing how we analyze frequency depends on our reference. So if you consider these black contacts in the stepped electrode here, that's implanted in the patient, brain parenchyma, some may be within the white matter, which is W, or gray matter in G. And if you use common average referencing, these signals may look very different compared to when you use bipolar referencing, which is basically subtracting the neural activity between contacts. So all the data that I show you now, I'm using bipolar referencing to give the most localized differences within brain regions. So for each raw voltage traces, when the patient sees a picture of an image, you can average all of these activity across time and analyze the different frequencies of interest. And this is a sample graph on one patient who saw 1,500 images. Here, I show you 4 different electrode contacts and the high frequency bands in the lateral occipital lobe. And in 2 of these contacts on the left, you can see here, there's not much activity in terms of whether the patient saw pictures of people, animals, or other images. But if you look at the 2 electrode contacts on the right, over here, you can see that I was able to find some regions in which seeing pictures of people resulted in a sharp high frequency band increase after the patient saw images of people or faces. And below on the right, you can see another contact shows higher activity when seeing pictures of animals. And there's numerous ways to calculate this, and we check with other methods to see if we get similar results. So now we're able to replicate to some extent some image category selectivity in early visual areas. So if we can replicate that in early visual areas, then not only can they differentiate categories, but can they differentiate between recognizing something and show evidence that we can record some sort of memory trace around these areas too. So one method to look for memory is to see if there's something called repetition suppression. So when someone looks at a picture for the first time, and then sees it again for the second time, you may see a decrease in these neural response the second time compared to the first. So this paper finds repetition suppression in the ventral temporal lobe, but my question was, can we see these similar signatures of memory in the occipital cortex? And no one really has provided any convincing evidence of memory signals in the occipital lobe or early visual areas, and whether there are memory signals before the signal gets to the hippocampus. So repetition suppression is still not really completely understood. And there may be other theories of why this happens, including like facilitation or prediction error. So it's very important for a test to be tailored carefully to correlate the patient's memory behavior. So Dr. Hermes has a data set using it's called the natural scene database, showing 1,500 images. But again, this test is not really specifically tailored to test for memory. The patients don't tell us whether they remembered an image, but I thought of a way to analyze this within their data. So this test shows 150 images per run, and there are 10 runs in total. But within these 10 runs, some of these images are repeated throughout the task. So for example, I may show you a picture of a smiley face in run 1, and then in run 3 and run 4 and so on. That's spread apart within maybe hours or days. So I coded a structure to analyze these repeats. So I can mark for the first time when a patient sees an image, let's say image 1, image 2, and image 3. And then the second time they see the image, and then the third time they see it, etc. And then this is just a simple bar graph showing that the distribution of when the image was shown, there's no weird outliers. So using the lateral occipital cortex electrodes, I averaged the signals across all specific repeats. And trial 1 would be the first time the patient sees an image. Trial 2 would be the second time, and so on and so forth. So when you average these high frequency bands across the first time the patient saw the image, and then compare that to the repetition trials like 2 to 6, you can see some of this repetition suppression within the electrode in the occipital cortex. But this is not really a fair comparison because you're comparing one trial versus two, the second time you repeat it or the sixth time, and combining it all together. So how can we analyze this data? So Dr. Hermes and team recommended something called a canonical response. So you can model a certain response across all trials, and it allows me to compare trial number 1 to trial number 2 to 6, eliminating that inconsistent comparison that I was doing. And again, with the theory of repetition suppression, I hypothesized that, the first time the image is shown of a panda, let's say, it will give me a normal response. And each of these subsequent responses that you see, I would imagine that they would decrease over time. And these are, again, very preliminary plots. And we do see some interesting evidence of suppression. And not only that, once the signals were shown the first time, the repetition trials, the first time it was repeated, the signal goes down, but it remains down no matter how far apart these images were shown. These are all very interesting signals. And not only that too, if you see this plot, I know this is a little bit confusing, but these blue lines represent the images that were suppressed multiple times when they were repeated. But you can see this red line over here that shows that there are some instances where when you see the image the second time, you actually see an enhancement of response. And we're still trying to figure that out what that means, because this has never been really described before. So again, the previous data set that I was working with isn't really designed for memory, and we don't really know for sure whether the patient remembered those images. So we can only infer with the best sort of analyses that we can do. So here I use the Newell test, which I'm familiar with from my previous work. And this test is great because with each repeat image, the patient tells us how confident they are in their memory that they remember this. So I decided to run this test on our patients here at Mayo and added some modifications to the original task. So I'd like to give special thanks to my prior mentors, Dr. Brunner and Dr. Hermes' lab. And these types of studies, you have to give like the highest standards of ethics, given that we're really recording from one of the most sensitive organs in humans. So I ask consent multiple times throughout, review the imaging carefully, and make sure I plan out and note the electrode positions carefully. And you need to really work closely with the epilepsy team, EEG technicians, and the nurses. So this is some of the pictures of the setup here. While Dr. Graywall implants these electrodes, I make sure they're connected in the right leads, and sometimes it changes. So you got to be ready to change your positions in terms of noting. You get the imaging on post-op day 0 and try to co-register them so you know which contacts you're looking at. You don't really record the post-op day 1, but make sure you check the clinical system to make sure you're getting the correct signals. You don't want to interfere with any of the clinical recordings that they do. And this is the setup and protocol. This is how it looks like. There's like leads coming out. And you could see I actually made this from Home Depot with the setup where you can actually hang all of the equipment in once. There's some of the connections here. Before starting the experiment, you got to make sure you're getting good signals. On the left, you can see very noisy signals. Looks like actually a scalp EEG. But if you actually, you can do this in a way where you can change the reference electrodes to get better and cleaner signals, like you see on the right side. So again, you work closely with the nurses, make sure before you start the experiment, you let them know how long it'll take, because sometimes these medications can be due and you can't have interruptions during the recording. After the experiment, you make sure you put away the equipment properly. So you can process this data while you're doing the experiment. Basically, you extract the EEG data, have the co-registered image to look at contacts of interest and analyze them with MATLAB. So after each session, I just check how patient performed in behavior. So these graphs just show 6 different runs as an example. Sometimes the patients can perform the test with about 60% accuracy, but this doesn't mean that patient only remembered 60% of the images. By measuring the ROC curves, showing these skewed ROC slopes, you know that the patient performed very well in terms of like being able to differentiate how confident they were with memory. On most of the patients we had very good behavior. And these are some of the figures you see during pre-processing steps with the raw voltage traces. And you need to correct them in multiple layers and eliminate noise. There's many graphs to check whether you obtain the good signals, and you have to eliminate the bad ones. And this is an example of each individual contacts in the y-axis and the average voltage traces in time, which is the x-axis. So you can see some of these hotspots in yellow here that kind of reflects how the voltage traces deflected in one direction, while the blue means the opposite direction. And time 0 represents when the image was shown. And with each of these contacts, you're able to see some channels with activity and correlate this with imaging to see the region of interest. And again, these are just preliminary figures. In the ventral temporal cortex circled here, you can see some signal differences in categories of images. And you can see some notable signal differences when the patient saw images of cars, for, let's say, in a region just lateral to the hippocampus. Here's another example of different categories. Now this one is the graphs that I'm actually interested in. So new on the right side here means the first time the patient saw the image, and then the old represents the second time the patient saw an image. And you can see some interesting just raw voltage changes here in the new, and then in the old session here, you don't see this sort of a hotspot here. So you know that you're detecting some sort of difference. Here's another example of the similar region in another patient. So yeah, I'm still working on how to extract clean high frequency signals within those regions and try to see if there are differences within them. And I'd like to thank everybody here at Mayo Clinic for allowing me to do this research. Thank you very much.
James, that was a great talk. You know, I think if you had to distill this down into like a 30-minute or 30-second elevator pitch, what would you tell us you're actually doing, and what's the goal of this?
So the goal of this is to really see where the earliest traces of memory can be seen, because we know in the hippocampus, there's been multiple studies where you see memory signals, but we don't really know how it goes from the visual cortex to the hippocampus. And I really want to detect where that earliest signal changes, and maybe that allows us to really target an area to perhaps enhance memory through stimulation and look for other sort of treatment algorithms to help people restore their memory.
Where do you hypothesize that area is?
It's hard to say for now, but I would say the lateral occipital cortex, which would be very interesting.
We want to open it up for questions because I can go for days. So
I know, if I can go, hey, James, congratulations, very interesting. I'm interested in looking at like how, machines will interpret some of this data. So it's nice to see how humans actually do it. But I have a question. When I briefly look at some of your slides, I had the impression that, and I wonder if that's because of evolutionary, changes, that apparently the signals on human and animals had a very peculiar intensity when compared to the other images. I wonder if you had a chance to try to even see if the face is actually one of the aspects that have an impact on that signal for face recognition, and the difference in the patterns on that signal as well. Because I wonder if you were to look just at the faces of somebody, and if you have a human without a body, I wonder if that signal is going to be very different as well.
Yeah, that's a really good point that you bring up. A lot of these studies, some papers only just look at faces, but from my previous work too, whether they see an entire, body of a human, but the face is kind of blurred out, you could still see some similar signals. So I think there's different sort of processing going on in terms of like face recognition versus just recognizing that that's a person right there. So that's still the ongoing work. Yeah.
Fascinating work, James. I had a question for you. So you talked about it, sort of repetition and suppression when you show these images over time, and each time we look at a picture, we may appreciate different elements of a picture, right? So the one you showed with the zebra, there's a little bit of a background, so maybe you focus on the zebra the first time and then you focus on the background the second time, or any picture, every time you look at it you may see a different element. How do you account for the differences in the focus of that particular image?
I didn't know we had so many functional neurosurgeons here, but that's a really good question. So a lot of these papers don't take into account for that, but through different types of images, you can average it across time with multiple trials. And that's how you kind of try to eliminate that. But what you bring up is also a good point, because let's say you see a picture of a zebra, right? And that has different contrast. So that's something that it's really difficult to account for contrast. And the only way to do that is just show different kind of animals or different sort of resolution. So you just keep repeating it over and over again. Yeah.
Can we open this up to it's cutting it out, I don't know why. This is fun. Hugo, or you have any questions from a research standpoint?
James, I see an opportunity right here, just based on some of the work that is being done already to potentially overlap with you and Sanjit and the work that you guys are doing, just to consider, and maybe make a comment as to where do you see chronic implants, like the wireless technology for which we already put intellectual property, both to record and stimulate both at the surface of the brain, and also we put the same intellectual property with wires and depth electrodes to be able to record. Where do you think and you can envision this moving in the future?
Yeah, that'll be very interesting. As we know, like ventral temporal cortex for human face recognition, for the patients to recognize their family members, for Alzheimer's disease, that may be a sort of a possibility, or just the recognition memory in general. Hopefully the lateral occipital cortex that I show here. There was a was a New England Journal paper a while back by Freed that showed stimulation of the hippocampus that improved memory in terms of like being able to go around places. They just use like a video game to go around places, and people who were stimulated in the entorhinal area actually had improved memory. So I think it depends on where you want to stimulate and what type of memory you want to enhance, but that's still a lot of work in progress, but it's very promising in terms of stimulation.
To remind me of a case, Sanjeet, that Dave Sapsovitz and myself and JP did a few months ago. Actually, we did a redo surgery on the same patient. We did the surgery a year ago in the lateral aspect of the temporal, occipital cortex inferiorly. And the patient did great. It was an awake craniotomy. And then after that, he said that he had difficult time recognizing faces and they looked distorted. And it was not in the cortical area. It was more in the deep, subcortical white matter tracts recognition. So eventually he recovered. The second time around, we had to make sure that we did the surgery. That was a few weeks ago. And we were touching those areas. So there's clearly something in those areas, in those pathways.
Yeah, that's a very interesting point too that you bring up. So there's also some stimulation right versus left ventral temporal areas. I think it was the right side where if you stimulate, you see distortion of faces, where if you stimulate the left, you don't really see that effect. So I think the laterality of where you stimulate for, future therapies like that would be very interesting to see.
And then the second thing that you remind me, we do know that there is a connection between the lateral entorhinal cortex and the nucleus accumbens. It is monosynaptic and it is mu-opioid dependent, because a lot of the times when, at least in animals, they get addicted to drugs based on the, both a combination of memory, drugs, but also visual input. And I wonder if there's something. I know that you didn't think that you had a lot of functional neurosurgery, but that was actually my very first paper that I published, back when I was an undergraduate at UC Berkeley, being monosynaptic, being mu-opioid dependent with that neurotransmitter. Any thoughts as to how this can also apply for drug-mediated reward and addiction to drugs?
Yeah, I can imagine, let's say I quit smoking and then I see a friend smoking, I want to smoke again too. So I think there's a lot of different sort of inputs that really enhance sort of addiction, things like that. I think there's one guy at UCLA that's studying that, but still really nobody knows, because it's really rare to get these sort of patients. And we have a lot of them here at Florida, so.
Yeah, I think that was really important, James. I think the most difficult resource here is the electrophysiology from the patients, right? So the time they're giving up to do these tasks and the surgery they're going through to give us this information, which they're doing typically for their epilepsy. But from Dr. Q's standpoint, the biggest issue we have, Dr. Q, is the sampling bias, right? So we put these electrodes in, but you only get signal from where you're recording, right? So we can say we think it's lateral occipital cortex, but if we don't have something at the same time in the hippocampus and the contralateral hippocampus and the contralateral occipital cortex, you don't really know that signal is the first signal, right? You know that that's the first signal you recorded. And that's why doing these multi-site projects and doing large series where we have electrodes sampled across hundreds of patients is how we're trying to get a law of averages. And that goes to Ian's point as well, about how to try to get around this stuff.
Maybe an opportunity for you and James to venture into, electrical engineering. I'm sure, I look at the work that Eddie has been doing. I look at the work that Kai Miller did in multiple channel record, but I wonder if there's an opportunity to create even more. Imagine doing patch clamping at the cortical level when you are in the operating room with a technology that is wireless, that you just put on the brain, you record as you're doing the surgery, or you record as you're doing your testing, and you're recording from single neurons and many thousands of them. Remember, we have built, even in this area that you expose, you have billions of neurons already, but if you just record from a few hundred simultaneously or a few thousand, I think that may, without injuring the brain, that may give you answers that no one else is able to, elucidate.
Yeah, I
You know,
think James is going to come to you with a money request for that here soon. So you're thinking along our lines.
Yeah, more money the better, but we'll see.
James, Dave Miller, you know, from the perspective of, I mean, there may be an opportunity. And have you thought about, we have a large population of stroke patients, many with lesions in certain areas. You know, I wonder, especially as we're getting more advanced with being able to image things and determine what areas are truly functioning, not functioning, affected, not affected. Would there be an opportunity to sort of, you know, you say that you're trying to determine where some of these areas are. We probably have patients in which specific areas are affected by their stroke and therefore out, so that you can map the fact that these are out and you can look at the other areas. And there may be an opportunity in some of these stroke patients that you can map their initial deficits, and then start to use some of your stimulatory stuff to see where things go. I mean, there's going to be some issues with obviously collateralization, reprocessing, remapping that the brain does in the setting of stroke. But have you thought about that opportunity of taking, because you know in animals we create lesions. In our stroke patients, we've essentially done that.
Yeah, exactly. That's a really good point too. I think me and Dr. Graywall also discussed this. It's a very low-hanging fruit study that would be very helpful. Basically bringing in stroke patients, maybe like a week off or seeing them in clinic and running some of these tasks would be very easy to do. And I think it'll be very interesting to see if it correlates with some of these electrophysiology studies. But yeah, that's a very, very good sort of large project that one person can do. All right.
Last comment, of course, in regards to money, the federal government, the NIH and DOD has tons of money. You just have to come up with a hypothesis to test it and apply for it. I look forward to reading the AIMS page.
That's Dr. Lee right there.
I'll do my best, but yeah, thank you.
Wonderful. Thanks, everybody. Sanjeet, thank you for leading the discussion. And Steve, thank you for presenting the updates. James, thank you for your presentation today. And Ali, I know you're still out there working. Thank you for joining us from UF as our residents are rotating. Everybody, have a great week. Thank you.